Hello everyone, thank you so much for joining me in the lab today. And today we are performing Exploring Creation with General Science, Experiment 13.1, Seeing a Part of the Digestive Process. The procedure for this experiment can be found in Exploring Creation with General Science by Wile, published in 2008. And the purpose of this experiment is to observe two different kinds of digestion: mechanical digestion and chemical digestion. The equipment and materials used in this experiment include a saltine cracker, a tablespoon, iodine, available at most drugstores in the first aid section, 2 small glasses. Juice glasses are fine, I'm using beakers here. We'll also need some water. It's not listed in your materials list, but you will need it, as well as a eyedropper or pipette in order to get the iodine out of the bottle. Also, I find that 2 stirring spoons are helpful, as well as your eye protection and a pair of gloves. So you're going to go ahead and put on your eye protection as well as your gloves. Also, I find that an apron might be a good thing to have in this experiment because the iodine is staining. So if you get any on your clothes, you are going to have a stain in your clothing. So wear an apron as well. Okay, step 1 of the procedure tells us to break the saltine cracker in half. And then we're going to crumble half of the cracker and allow the crumbs to fall into one of the 2 beakers. This entire cracker is crumbly. Okay, so this is really our mechanical digestion that we're exploring here. Here we're just breaking up the whole cracker, or a half in this instance, mechanically, kind of like the teeth would do in your mouth without the addition of any saliva. Step 2 tells us to add 2 tablespoons of water to the cracker crumbles and swirl the glass to mix the cracker crumbs with the water. Wow, that's a mouthful to say. So here's my tablespoon and my water. We're going to add 2 tablespoons of water. 1, 2. And we're going to swirl that. Step 3 tells us chew the other half of the cracker really well. Do not swallow, but spend a lot more time chewing the cracker than you normally would before swallowing. All right, here goes. Don't need to see this on camera, but I will be chewing the cracker. Now, I'm not swallowing, and yes, I am talking with my mouth full, but I'm going to spit the stuff in my mouth out into the other glass. Okay, it's gross, as the text says, but science is gross sometimes, and deal with it. So, when I spit this into the beaker off camera, you will see this bolus of food in here that has come from my mouth. So, I'm going to apologize in advance for that, but there you go. Lovely. And there it is in the glass. Okay, now we're going to add 2 tablespoons of water to that glass as well. Okay, and we're going to go ahead and swirl that glass and see if we see any differences between the two. Okay, so this is the first beaker where I just crumbled up the cracker. That's exhibiting mechanical digestion. And in this one, I actually chewed it and then put it into the beaker. And this one had the addition of my saliva, of course. And so that is exhibiting both mechanical as well as chemical digestion. Okay, so you can see the cracker bits in this one are quite a bit smaller. The water is getting quite cloudy. And this one, the cracker bits are bigger, and that water is still mostly clear. So go ahead and record those observations. Now, step 7 tells us to add equal amounts of iodine to both glasses. It doesn't need to be a lot, just a drop or 2. So I'm going to go ahead and add 2 drops of iodine to each beaker. Now, when you add the iodine, I recommend that you work over paper towel or paper plate because the iodine can stain your countertop as well. Remember that you want to make your hypothesis before you do this, knowing what you know about iodine from previous experiments, as well as your background research. The iodine is an indicator stain for the presence of carbohydrates. Okay, so we're going to go ahead and put 2 drops in each. We're going to swirl each glass and get them under the camera here so you can see them well. So already you can see a staining. This very dark blue to dark purple to dark black is the indication that we have carbohydrate present. And in this left beaker, that is where we had the mechanical, also called physical digestion, and the chemical digestion going on. And in this right side is where we had just the mechanical or physical digestion. So if you can't appreciate it from the camera, this beaker that is showing both the chemical and physical mechanical digestion is darker than the one with just the physical/mechanical digestion. And we still, of course, have bits of cracker here in the one where it did not get chewed. Okay, so go ahead and record your results, and don't forget that you want to write up your discussion and conclusion and reference after doing so in order to complete your laboratory journal entry. Thank you so much for joining me in the laboratory today, and I look forward to seeing you next time.